if a cell gets a green light, like we see here, during G1, it's probably going to totally divide. So it's probably going to go all the way through the cycle from there. And the other checkpoints are probably not going to stop it either. So if a cell goes ahead through the restriction point, we probably see cell division in its entirety. However, if a cell gets stopped at the restriction point, then the cell moves into what we call G0, or sometimes G0. Right? This G0 phase is a non-dividing phase, which means the cell is not actively undergoing cell division. Don't be confused here that the cell's alive. It's not dead or anything like that. It's just not actively dividing. Most of the cells in your body are actually in G0 interphase. They're not at an actively dividing point. Some exceptions would be, like we mentioned earlier, your skin cells, but other cells are in G0. Some of those are going to stay in G0, like those nerve cells I mentioned. Others will be able to be called back out of G0 and divide if necessary. There we're talking about things like bone cells or liver cells. Basically any cell that we would need to turn on if something would need to heal or things like that, we can do that. I like to think of cells in your body like the military reserve. Right? Those men and women are going about their lives, doing their jobs, all those kind of things. But if we went into a time of war, they'd all be called back to active duty in the military. Most of your cells are in G0. They're doing their job, doing what they're supposed to do. But if we needed them to, we could call them back in to divide and make new cells. Now the control system at the molecular level involves a rhythmic fluctuation of molecules. So throughout the cycle, you've got some molecules that you are getting more of, and then less, and then more of, and then less, and so on. Now, a go-ahead signal at that molecular level usually means that some sort of molecule got phosphorylated, or we added phosphate to it. Alright, so one of these proteins involved in cell division probably gets phosphorylated, and that causes it to go ahead and do its next job. Phosphorylation is performed by enzymes called protein kinases. And we mentioned those during Unit 1. We're about to learn a lot more about them here very shortly, but these are the enzymes that perform phosphorylation. Now, one of the specific types of kinases that we see in a cell are called CDKs, or cyclin-dependent kinases. Now, these are specific kinases for the cell cycle, but they don't actually do this rhythmic thing that I mentioned. CDKs are there all the time, but in order for them to get activated, they have to bind to a protein called cyclin, hence the name, right? Cyclin-dependent kinase. So the only time that they actually show kinase enzymatic activity is when there is cyclin around. So we call them cyclin-dependent. And as you might guess by their name, the level of cyclins does fluctuate as we move through the cell cycle. Now you see that here in this picture, one example of this. If the level of a cyclin is high enough to activate the CDK, when we reach a checkpoint, then you get a go-ahead signal. If, however, on the other hand, the cell is not prepared to proceed, it will keep cyclin levels low, and the CDK won't activate, and you get a stop signal. Now there are tons of different CDKs and cyclins for the different checkpoints. I'm trying to think back to medical school. I think there's somewhere on the order of 20 to 25 different cyclins and CDKs that we've identified. Maybe the most important one and the first one that was discovered is called MPF, or maturation promoting factor. This is a cyclin CDK complex that takes part at the G2 checkpoint, so not the restriction point of G1, but at the G2 checkpoint. And as you notice here, if the cyclin level here in purple gets high enough, then you get activity of the cyclin-dependent kinase, MPF. So you'll notice here there's a rhythmic fluctuation where the activity of MPF is high right here at the end of G2 going into M phase. And so the cell can regulate that cyclin level if it needs to, and it can keep it from getting high if it wants to stop the cell cycle. Cyclin CDK complexes also might activate other enzymes that cause an event to happen. So it's not just that they start or they stop the cell cycle, but something like MPF also probably helps the nuclear envelope break down, the spindle to start forming, and the chromosomes to condense. All things that happen just before mitosis or during early prophase. So, like we said, MPF is at G2, so just before the cell is about to divide, as long as MPF is active, we're going to see these steps of prophase start to come to light. Now the question becomes, what causes a change in cyclin level? We say it fluctuates, but why? And the answer is two things. We have some internal factors that allow the cell to go or to stop, and we have some external factors that we're going to look at here in the next couple slides. At the internal level, a cell has to meet certain conditions in order for you to get active kinases. So what do we mean by this? When we approach the M phase checkpoint, the M phase checkpoint is sort of between metaphase and anaphase. Basically, cyclin levels will be held down until all the kinetochores, which are those things right at the center of a chromosome, right, get attached to the microtubules. So until all the chromosomes are lined up like they're supposed to be, we don't raise the cyclin level inside the cell. So that would be an example of an internal condition. If the kinetochores are not attached, you don't get the cyclins and you don't get passage through the M phase checkpoint. But if the internal conditions are right and all the kinetochores are attached as they're supposed to be, then we get that cyclin increase and we get our M phase checkpoint cleared. Now the external conditions are a few different things. The first one we often talk about is density-dependent inhibition. Remember, inhibition again means to stop or to slow. If a cell is touching other cells on all sides, it keeps its cyclin levels low so that we prevent division and we don't get overcrowding. We'll see a picture of this on the next slide here. Animal cells also have what's called anchorage dependence. Anchor, right? So a cell must be attached to a surface underneath of it in order for it to divide. In other words, you don't get cells just floating out in random places in your body and dividing. They have to be attached to something first, and we call that a substratum. We've got a picture of this coming up too. So here's anchorage dependence, right? If these cells are not attached to the bottom of this little glass dish that we have in this picture, then they will not divide. Then we have density-dependent inhibition here. Once we get a nice, smooth, full layer of cells where the cells are touching each other on all sides, we stop division. So that's why we call it inhibition. Now, if we were to take out a couple of these cells, then in a normal situation, like you see here, the cell division will be turned on right around this opening, and we'll fill back in that gap until we get our nice smooth layer of cells. Something that we're going to talk about here in just a minute is cancer, though. And in cancer, you don't have density-dependent inhibition. So instead of getting that nice layer of cells and stopping, you get this massive buildup of cells all on top of each other. And so we'll come back to cancer here in just a minute and talk about that some more. Now another external factor, sorry, I overlapped this here, is growth factors. Growth factors are proteins that can be released by certain cells, sort of like hormones, that will stimulate other cells to divide. Now we've got over 50 different growth factors for humans. One of those being HGH, or human growth hormone, that you hear about sometimes with abuse by professional athletes and things. But cells are going to respond differently to different growth factors and also different combinations of growth factors. One of the growth factors that seems to have appeared on several of the previous AP exams over the last few years is called PDGF, or platelet-derived growth factor. Platelet-derived indicates that this growth factor comes from platelets, right, which are actually cell fragments in your blood. We might be familiar with platelets because they're the things that help you form a clot when you want to, right? So if you cut yourself, platelets will help you to clot that blood so that you don't bleed forever. Now platelets only do part of that job. the guys that are really responsible for closing up that wound and stopping you from bleeding are cells called fibroblasts, which you can see up here in this picture. Fibroblasts have receptors for this platelet-derived growth factor. And when those receptors get bound by platelet-derived growth factor, fibroblasts are sent through that G1 restriction point and are told to divide. So, if you cut yourself, the platelets in your bloodstream will release a bunch of PDGF as soon as that injury occurs. This PDGF will stimulate fibroblasts to multiply and heal up your wound, and in the process stop your bleeding as well. And so platelet-derived growth factor is essential for the healing of injuries. Now, we do want to spend a few minutes to wrap up here with cancer. Like I said, cancer is not a huge deal on the AP exam. In fact, I don't think there was anything on last year's exam, but it's been on there before. And the College Board does say that you're responsible for understanding cancer. So we're going to talk about it a little bit today and a tiny bit when we talk about genetics, and I think you'll be good for that. Essentially, a cancer cell does not pay attention to the normal signals that stop or regulate the cell cycle. They don't need growth factors. They don't need any kind of stimulation. They just constantly go through cell division. Cancer cells also will avoid apoptosis. So this is a way that your body can kill cells it doesn't need anymore or that start to go bad. Cancer somehow avoids apoptosis. And so cancer cells just grow uncontrollably. Now, when a cell is cancerous, that means that it's got a change in one or more genes. So its DNA is messed up. You know, DNA leads to proteins, so that's going to alter the function and structure of our proteins, and it's a lot of those proteins that help control the cell cycle. So if we stop making a protein or we mess up a protein, we may no longer have those checkpoints in place, and the cell can divide as much as it wants. Normal cells stop dividing, age a little bit, and then die after they've divided 20 to 50 times, depending on what type of cell we're looking at. This is the normal course of events in the body. Some cells die a lot quicker than others. If that's too soon, they get replaced. But cancer cells can be immortal. There is a culture in a lab, I think somewhere in the Carolinas, where they have been culturing cancer cells called HeLa cells since 1951. And they continue to grow and reproduce. I don't know if anybody's ever heard of Henrietta Lacks. There's a really interesting book about her life now. Henrietta Lacks was an individual who had cancer, and they extracted these cells from her and began culturing them in a lab back in 1951. Like I said, today these cancer cells are still dividing. Now, cells that are transformed in your body usually get identified and destroyed. Your body has a great defense system that we'll learn about toward the end of the year, and that defense system usually notices cancer cells and kills them. This is happening in every one of us every day. You are having cellular changes called mutations, and you're none the worse for it because your body identifies those mutations and kills them. You know, this is kind of scary to think about, but if we have an intruder at school, most likely we're going to be able to identify that intruder quickly. He's not going to be one of our students, and notice I said he, because usually those individuals are males. However, if one of our students came to school one day and intended to do something bad, they would get into the school just fine, and we wouldn't notice them. This is a problem in your body as well, because cancer cells come from your cells. It's not like an invader, like a bacteria or virus that we would notice so easily. Cancer cells are where your cells turn bad, and so they don't have a problem getting in there. And so for some people, particularly as you get older and older, those mutations that we get can get overlooked and not destroyed, and then they multiply. And so if a cell survives, it can divide, and it forms a mass of cells called a tumor. Now, a lot of times we hear tumor, we think bad, but that's not necessarily true. A tumor is just a mass of cells, and so some tumors can be what are called benign tumors. This is an abnormal cell that's growing out of control, but it stays where it starts, and it doesn't really have a lot of changes going A benign tumor is just a mass of cells, and so these can often be surgically removed